Good afternoon, everybody. Welcome to a Wednesday overcast afternoon of Chem 170 with your host, me, Dr. White. Can't believe this semester is just about over. Time flies when you're having fun with organic chemistry and Dr. White. I hope it did for you. It did for me. All right, important things. First of all, when I'm through today, which is going to be an early lecture, we're going to end early. I will send out an email with the password for test 4. You'll have until tomorrow. I don't know, I think 4 or whatever I put down. You'll see when I send out the email to take test 4, upload your answer as a single PDF file to D2L. All right, other important thing. Don't forget today's the deadline for the extra credit project. I think I give you till midnight tonight to hand it in. Make sure you do. It's 10 bonus points. And finally, a week from this Monday will be the final exam. By Monday, well, first of all, for test 4, by Sunday or earlier, hopefully earlier, I will post your test scores for test 4 and D2L. And by Sunday or earlier, hopefully earlier, I will send you each an individual email with the points for each answer on test 4. Now, back to the final. A week from this Monday, I will be giving the final. A week from this Monday, I don't know what date it is, I can look it up, but I better. Hold on while I do this. And that would be December 12th. On December 12th at 1 p.m., we won't have a class, we'll have the final. I will send out the email for the final exam. You'll have until next day, whatever time I put down, to upload it with your answers. Now, the best way to study for the final exam is use all your hourly exams as practice problems. Excuse me. I'll say that again. The best way to study for the final is use your hourly exams as practice problems. Am I going to put the same problems? No, but look at the practice problems you've used all semester. They've been similar to the problems on the test. That's why I call them practice problems. And the same thing for the final exam. Now, by Monday, before our class, I'll post and also send out as an email the point breakdown for the final exam. I'll have it broken down 2 ways. One, how many points for general knowledge, how many points for nomenclature, how many points for reactions. I'll also have it broken down how many points for the material from test 1, 2, 3, 4, and the new stuff, amino acids. Now, the way I have my final set up, the first part is from test 1, second test 2, then 3, 4, new material at the end. And I don't know, I think it makes it easier for you to take the final. I could play shuffle all the information like shuffling a deck of cards. Why bother? It helps you if they're all separated. All right, so more also I'll talk about more next week about finals week. And any questions on that? All right, let's go back and review something. And you should see on your screen chapter 17, amino acids. Now, I don't have a problem set for this chapter. Oh, no, why not? Well, there's not much to ask. First of all, you should know how to draw an amino acid. And an amino acid is, and I have one way of drawing it, is a carbonyl with a hydroxyl group, then CHR, this is the alpha carbon, the carbon attached to the carbonyl carbon. On that, it has an NH2 group, and this is an amino acid. Now, if R is not hydrogen, like a methyl, ethyl, or whatever you want to put down there, benzene ring or whatever, then the alpha carbon has 4 different groups. Carboxylic acid, R is not hydrogen, the amino group and hydrogen, which makes this a chiral carbon, which you should know. Next, and I'm not going to ask anything on Twitter. Oh, I did something earlier class today that I'll do now. Hopefully you enjoy this. One of my big hobbies is electronics, building stuff. And this morning, instead of going click, the switch is off, I went like this. Click, the switch is off. I thought that was cute. Then again, sometimes my humor is, well, different. And I like that. Maybe you don't, but I do. I tell you, my class is a democratic dictatorship. I let students vote and then I decide. But anyways, moving on, Zwitterion I'm not going to have on a final or a test. And this is for the final, this won't be on. But what you should know is that if you take 2 amino acids, they form an amide bond. And the general reaction, which you learned for test 3, carboxylic acid, an amine where R prime and R double prime can be hydrogen, and you form an amide. In this case, this is my R double prime, and this whole thing right here is my R group, or you could switch it. And one of the hydrogens is the hydrogen, and you form an amide. And you should know that I'm not going to ask what's a peptide bond or what's a peptide, but proteins are formed by combining 50 or more amino acids. So if I have, say, 60 amino acids, and Mother Nature uses a catalyst called an enzyme, you'll make a protein. If I can spell it. Did I tell you I was always the first one down in the spelling bee? Still would be. And that's how Mother Nature makes that. And your skin is a protein, your hair and everything. Now, when you eat something that has protein in it, and it goes down to your stomach, and also, I forgot which intestine also breaks it down, you have acid and water. And one of the miracles of life, I always feel, is we all have cells that make hydrochloric acid, HCl. And you learn for test number 3, that if you take an amide, react it with acid and water, you get back the carboxylic acid you would have used to make that amide, plus the amine salt, because remember, amines are basic, I have a bad hair day. I'm having a bad hydrogen day. Oh no. Or hydrogen writing day. Hopefully the hydrogens in my body are doing okay. And that's how your body breaks down protein to amino acids. And an amino acid, these two are connected. And here I show you the example of that. And that's why I don't have, because what can I ask you? Well, I could also ask one other question. If you take a protein and react it with acid and water, what do you get? Amino acids. And it's amazing. Most people, when they say, I hate organic chemistry, they don't realize all the stuff that's going on in their stomach and other parts of their body. Just growing hair is organic chemistry. What's left of mine? All right, any questions about amino acids and proteins chapter? Now you see why I don't have a problem set for that. Nothing like tea during the day from the Republic of Tea. All right, let's go through some important things you should know leaving my class this semester. By the way, the amino acids and proteins will not be on test number 4, but a few points will be on the final exam. And it's subtle Wednesday. Don't you like it when I'm subtle? Get the hint? Know this. And it works. All right. 3 things you should know leaving my class. One is, how do you describe with words and a general reaction if you eat something with a fat or oil? And in your stomach, there's acid, there's water, so we call that hydrolysis. And what's the key functional group in a fat or an oil? An ester. And what it is acid hydrolysis of an ester, and the general reaction you learn for test number 3. You take an ester, react it with acid and water, you get back the carboxylic acid plus the alcohol you would use to make that ester. So you should know when you eat a fat or oil, how to describe it with words, acid hydrolysis of an ester, how to describe it with a general reaction, ester. Acid and water, you get back the carboxylic acid plus the alcohol you would have used to make that ester. Oh, that's why I was running out of room. I forgot to go to my whiteboard. Hold on, let's try an experiment. And it worked. All right, now I'm on my whiteboard. All right, the next thing you should know is how to describe with words and a general reaction when you eat a carbohydrate? And what's in your stomach? Acid. And there's water, and we call that hydrolysis. And you learned, and hopefully for today's test, you learned what's the functional group that holds the carbohydrate together, the glucose molecules, and that's an acetal. And so, how do you describe that with words? It's acid hydrolysis of an acetal. And for test number 2, you learned if you take an acetal or ketal, R prime is H, it's an acetal, R prime is carbon, it's ketal, acid and water. Isn't it amazing all the stuff the acid in your stomach with water helps you do? And you get back, keep your eye on the carbon with the 2 oxygens. That becomes this carbonyl carbon, carbon double bond to oxygen. And what was attached to it, R prime or hydrogen, R double prime. And you also get the alcohol you would have used to make that acetal or acetal or ketal. Now let's see how small I can write the number 2. That's pretty small. If you want to balance it, you can. You don't have to. All right. So when you eat a carbohydrate, you should be able to describe with words what happens when you eat a carbohydrate. And that is acid hydrolysis of an acetal. You should also know how to write the general reaction. And that's acetal or ketal, acid and water. You get the ketone or aldehyde and the alcohol you would have used to make that acetal. This case is glucose, and both are in the same molecule, which I showed you. And the final thing you just learned, not yesterday, Monday, and I just went through it again. How do you describe with words and a general reaction when you eat a protein? And what's the functional group that holds amino acids together? Don't use peptide. I'll mark that wrong. I'm an organic chemist, and we call it an amide. So in your stomach, there's acid, water, so we call that hydrolysis. And the key functional group in a protein is an amide. So this is how you describe with words when you eat a protein. Acid hydrolysis of an amide, because the key functional group holding amino acids together in a protein is an amide. And the general reaction you learned for test number 3 if you take an amide, and you don't have to put R prime and R double prime can be hydrogen. I do, but you don't. And what you get back is the carboxylic acid you would have used to make that amide, plus the amine salt of the amine you would have used to make that amide. Like I said, I'm having an awful hydrogen drawing day. My hair's okay. Just I can't. Is this Friday the 13th? Oh no, we don't have class on Friday. And that's what you get. So once again, leaving my class. See, it says hint, know this. You believe everything you see on the screen, don't you? Oh, that was awful. But anyways, leaving my class, you should know how to describe with words and a general reaction when you eat a fat or oil. That's acid hydrolysis of an ester. And the general reaction is ester, acid and water. You get back the carboxylic acid and the alcohol you would have used to make that ester. When you eat a carbohydrate, what's the key functional group holding the monosaccharides? Took me a second to think about that because it's a word I don't use that often. It's the area organic chemistry I don't do much in professionally. If you eat a carbohydrate, what's the key functional group that holds glucose together, molecules of it in a carbohydrate? An acetal. And in your stomach, there's acid and water, and described with words is acid hydrolysis of an acetal. And for test 2, you learned acetal, ketal, acid and water. You get back the ketone or aldehyde plus the alcohol you would have used to make that acetal or ketal. And finally, eating a protein. What's the key functional group that holds amino acids together in a protein? It's an amide. Don't put down peptide or I'll mark it wrong. And the way you describe it is acid hydrolysis of an amide. And take an amide, react it with acid and water. You get back the carboxylic acid plus the amine salt of the amine you would have used to make that amide. All right, a few things I want to talk to you about, but I do want to say this. At this point in time, remember the first or second class, I think it was the first class, I said, in Chem 170, you'll find out my world of organic chemistry is really your world of organic chemistry. You just don't know it. And now I can say my world of organic chemistry is also your world of organic chemistry, and you know a lot about it. And hopefully that's a good feeling. You know more than most people on this planet now about organic chemistry. And hopefully that's a good feeling. All right, we're going to have a lab. But before we do the lab, and it's a quick lab, and we're going to get out really early today. But I thought I'd teach you a few quick things. One I might already have done, but the other I haven't. Did I teach you the quickest way to clean a bathtub and a shower stall? All right. The one job I detest in my house the worst is cleaning the bathtub. I mean, I hate it. Cleaning a toilet or anything like that doesn't bother me. Cleaning the bathtub, especially when it's got a lot of scum on there, I hate that with a passion, which means I let it go too long to the point is it's really bad, which makes me hate it worse. Well, a number of years ago, I was cleaning my bathtub, spending a half hour scrubbing things that are caked on because I didn't want to do it and I let it go too long. And all of a sudden I go, you're an organic chemist. How do you get this off quickly? And dawned on me, oh, I know how. And it worked. All right, now you probably heard you can use vinegar. And the vinegar you can get your hands on is too dilute, only 4%. Now, as you learned in one of our labs, what's bathtub scum? Well, if you use bar soap, and this will not be on a test, this is something you can use the rest of your life that makes your life easier. In the water in Chicagoland, a lot of others, we have calcium cations, ions, and you get an ion exchange reaction. And we did this in the lab. And this calcium carboxylate is much higher molecular weight, double almost or almost double than the starting soap. And that's what's causing the caking. It precipitates out and sticks to your bathtub and my walls. Not as bad as the bathtub, but anyways, or if you have a shower stall, I have one downstairs. I don't use it. I've got a nice shower in my bathtub upstairs in my home. So how do you get rid of it? An acid. And the acid I found that worked the best is oxalic acid, this right here. And again, switch is totally off. What happens is you get back tallow fatty acid, a mixture of stearic and oleic acid, which is more water soluble, breaks it down, plus calcium oxalate, which is a smaller molecular weight, and therefore more water soluble. Well, where do you get oxalic acid? Well, it turns out there's a nice product that I did a little research years ago and found it. I wasn't aware of it. All right, everybody see on your screen the barkeeper's friend? And barkeeper's friend ingredients is oxalic acid and soap and some, I think some, not sand, but some other abrasive, that's the word. And if you notice right here on your screen, warning contains oxalic acid. And let's see if I go like this. And what's in there is feldspar, which is the abrasive oxalic acid. I didn't know there was citric acid and surfactants, which means soap. And here's what I learned how to do. Your bathtub and walls are caked up. I have one of those removable shower stalls heads, and I take it, turn on hot water, and real quickly spray down the walls just to get them wet with hot water and the bathtub. Then I take the powder, they make a liquid, but I like the powder, and I'll sprinkle it all over on the walls. And then don't use a sponge, use good high quality pieces of paper towel that's slightly moist, and you just make a paste over everything. My walls and bathtub, that takes about 3 to 4 minutes. Then you let it sit for 15 minutes and you're not doing anything, scrubbing while you're sitting. And you come back and with a wet paper towel or one of those 3M greens pads, I learned from my sister, cut them in quarters and they last longer. You don't have to waste a whole sheet. And you moisten it and you just lightly rub it and almost everything comes off immediately. Then I take my removable shower stall and wash it down. My actual manual labor time it takes me, or I'm actually working, instead of taking an hour, 45 minutes scrubbing your brains, really hard scrubbing, it's about 10 minutes or less. And most of that is very light just rubbing. And it makes my life easier. And I don't let it go as long, even though I still let it go because I still hate it, but it's a lot easier. Also, that same product works great on pots and pans that have stuff gunked on it. That's a really nice professional term. I like to cook. I've been cooking for many years, and so I have all clad stainless. But even before, And one of the things I learned was you have things where you're frying and it's stuck on the bottom of the frying pan. After you've gotten all the grease out of there, with a spoon or whatever. And then after it's cooled down, you put in some water, turn on the burner, get it boiling, and with your spatula, most of the stuff will come right off. You just rub it a little. It's called deglazing a pan. And then if there's still stuff on there, you put a little hot water in there from your sink, dump it so it's just moist. Barkeeper's friend, rub it, let it sit 2 minutes, and rub it again with those 3M pads, and it comes right off. I remember years ago with the 3M SOS scouring pads, you'd be sitting there for 10, 15 minutes. All right, next thing. Did I teach you, and I don't think I did, how to unclog a sink in the kitchen or bath? All right, in the bathtub. All right. Years ago, when I bought this house, I moved in, and within about 3 weeks after I moved in, my kitchen sink clogged up. I figure, all right, new house, I'll call up the Roto-Rooter guy. I don't know which company that does roto-rooting I got. And he came out when their people came out, put their Roto-Rooter down my sink, cleaned it out, and said, it's clean. And I saw, yeah, the water's going down. And he said, here's our 6-month guarantee. If it clogs up, call me. We'll come out. We won't charge you. I said, oh, thank you. At this time, I was working at a chemical company where I was senior manager, and I was not hourly, salaried, and I was quite busy. And I'd have to stay home and wait for this guy to show up, and then I'd get to work late. Not late where I'm in trouble, but I missed time that I should be working. I had a lot to do. Well, within about 2 or 3 weeks, guess what? It clogged up again. So I called them up and said, one of the things I should point out, my father taught me this, and I realized he was right when I became an organic chemist. When you're cooking, never pour grease or fat down your sink because it will solidify and clog it. What I do is when it cools, or I'll take it out and put it into a Pyrex measuring cup, let it cool. Then I'll double bag it in Ziploc baggies and throw it in my garbage and wipe it with a paper towel. And that keeps it from getting clogged. Well, anyways, it's clogged again. I wasn't dumping stuff down. So I call him up and he says, all right, I'll be out. You're under warranty. He does it, apologizes. All right. 2 weeks later, it's clogged again. Now I'm getting upset. And I call him and he comes out, same guy. And now he says, well, I'm going to use this chemical too, stuff. And I look at the bottle, being a synthetic organic chemist who specializes in surfactants. And essentially all it was soap and some water. Well, a lot of help that did. Well, he cleared it. And within a week or so, it clogged up. I said, I'm wasting all this time. The money I didn't care about was my time where I needed to be somewhere else. So what did I do? Luckily, near my house, a 6, 7 minute drive away, there's a really good Home Depot. And it's been that way for many years. There's another Home Depot a little farther in the opposite direction. And it's not as good as the one I go to. This is near Schaumburg. But anyways, I go in there and they have a whole wall of drain cleaners. I pick up a couple, and being a chemist, I look at the back, I see hydrochloric acid. And this nice gentleman who works there comes up to me and says, can I help you? And I said, sure. My drain is clogged and I have a question for you. He said, all right, what? I said, once in a while, even Dr. White does smart things, and this was pretty smart for me. I said, all these drain cleaners, which one do the plumbers use? And almost this quick, he points that one. And I pick up the bottle and let me show you which is that one. And if you see on your screen, there's a bottle of Instant Power Hair and Grease drain opener. And this was in a big, heavy plastic Ziploc bag. And on the bag, it said, use eye protection when using this product. It's the only one that was like that. I look at the label, ingredients, and it has potassium hydroxide and sodium hydroxide, a base. And both acid and base will saponify the grease, break it up. But I figured, all right, let me try this. But before I left, I looked at the label and I said, oh, this is a base and those others are acids. They said, no, this is an acid too. And I told them, no, I've got a PhD in synthetic organic chemistry, and potassium hydroxide and sodium hydroxide are a base. He said, no, that's an acid. I wonder, all of a sudden the light went on in my head. Why am I arguing with somebody chemistry when he doesn't know what he's talking about? But he's been very helpful to me. So I said, thank you for your help and have a good day. And I bought it and it worked. What I do differently was, instead of waiting about 5-10 minutes like they say, I gave it a half hour. The other thing, in my kitchen, it takes a while for the hot water to get from my hot water heater in my utility room to my kitchen. So I went down to my utility room sink and filled up a bucket of real hot water. So when it says pour water down after the 15 minutes, I used hot water, because that would, any kind of reaction still going on, would help make it go faster. And guess what? Boom, it was clean as a whistle, I assume, because it was a good 12 years later when it was slightly clogged up, and I used this again. Now, I also have used this in my sink in my bathroom, but more importantly, my brother-in-law used it in his bathroom. A number of my students have used it in their tubs and also their sinks, and this stuff works real good. And when I've had plumbers out, they all agree that's the best stuff. And how does that do that? Well, it's base hydrolysis of an ester or base hydrolysis of an amide. And now I've told you 2 good things that you can use for the rest of your life. So what I would like to do, since the lab is pretty quick, let's go straight through. We'll be done in about 15 minutes or less, probably less. And I'll say goodbye. In a little while, I'll send out the password. And for those of you who know it, don't forget to use the method. All right, let's do the last lab. And make sure you've handed in labs. All right, the last lab is the carbohydrate lab. And this in face-to-face is a very quick lab. And so it's going to be quick now too. Now, what this really is, I should have named this, is the starch lab, but I call it carbohydrates. Well, it does deal with carbohydrates too. Now, when I draw a carbohydrate, you think of a straight string, but it's really a helix, the glucose molecules. And if you think about the glucose molecules, in a starch, and each glucose molecule has a lot of hydroxyl groups all along here. Now, back long time ago, before organic chemists had the electronic equipment we now have, which unfortunately I don't have time in a 1-semester course to teach you, like infrared spectroscopy, hydrogen nuclear magnetic spectroscopy, and also carbon nuclear magnetic spectroscopy. I've also done boron spectroscopy, nuclear magnetic. By the way, how many of you are familiar with what's it called, the imaging in hospitals? What's the right name? I always forget. Anybody know?
Are you talking about the MRI?
Yep. What was that?
Magnetic resonance imaging.
Yep. And guess where they got that from? Organic chemistry and uh, proton nuclear magnetic resonance. You're just flipping the spin on a hydrogen. And then someone came up to take that data and turn it into images, which was quite amazing. But anyways, before all that equipment, chemists were learning about carbohydrates and sugars and starches. And one of the things they were trying to find out, what foods have starch in it? How do you do that? Well, they came up with a visual test. And that test uses tincture of iodine. Now, if you have starch and you add a, say, potassium iodine, this is a solid at room temperature, starch and Ki interact, it's potassium iodine. This is a brown color or red brown. When the iodine gets in here and is associated with the hydroxyl groups, this now becomes a dark blue. So dark blue, if you look at it, you might even think it's black. So if you can use this, it will tell you, does it have a starch or not? Now there's a problem. Ki, potassium iodide, is not that soluble in water. I screwed up. Erase all this, nerp, nerp, let's back up. I got ahead of myself. Once in a while, I do mess up. Let's start again. I'm cutting this out of the video. Nah, I'll leave it in. Dr. White makes mistakes. Now you know I'm not a robot, I'm a human. If you take iodine, which is brown, and react it with a starch, or interact it, the iodine interacts with the hydroxyl groups, and there are a lot of them here, to again make this dark blue. Now, iodine is nonpolar. And you know light dissolves light, so that's not going to be soluble in water. That's also a solid. So what someone found out was if you take potassium iodide and react it with iodine, you get this potassium iodide Ki3, K plus I3 minus. And this is now polar, and it's water soluble. So you can make a solution of this. And in fact, this with a little alcohol is called tincture of iodine in water. And if you were my age, when we were little kids, if you got a cut or scrape, your mother would take you into the bathroom because most people kept it there. And there was a bottle of tincture of iodine, and they put it on the cut, and it burned like heck because of the alcohol in it, and it disinfected that area. And later, a company called Bactine came out with the first product that you could put to on a scrape or a cut to keep it from getting infected that didn't sting, and they made a lot of money. But back to this. Now here, again, if you take a starch, and react it with the tincture of iodine, which has this, and you've done this reaction to make this now water soluble, and what happens is the I3 gets in here with all the hydroxyl groups. And now same thing, this turns dark blue, so dark you might think it's black. So this is now a test. Does something have starch in it? And so in this experiment, you would be taking on what looks like a dish, we call it a recrystallization dish, certain items, and you would add a few drops of tincture of iodine, wait about 20 seconds, and is it brown, reddish brown, or dark blue? And let's take a look at this following video. Hold on. By the way, this stuff is really good. I keep a gallon bottle in my house. I don't use it that often, but it's got almost an infinite shelf life. It's inorganic. All right, time out for commercial.
We realized that maybe the key for making
an important constituent of our daily food are carbohydrates, which we consume in the form of starch. Starch is a high-calorie food that converts to sugar during digestion. Starch is also produced by all green plants as an energy store. Today we will conduct an experiment to test the presence of starch in potato, rice, sugar, and salt. For this, we will need iodine solution.
That's tincture of
which is
iodine.
brown in color. Note the change in the color of iodine as it is put on the rice and potato.
See how that looks almost black? It's really dark blue.
Both turn the iodine blue-black. Put some iodine on the sugar cubes. The color does not change. Salt too does not change its color. This proves that sugar and salt do not contain starch, while the starch in rice and potato reacts with iodine to form a blue-black compound. Now, the reaction is because the blue-black color is basically the formation of polyiodide chains from the reaction of starch and iodine.
All right, let's look at today's lab. What you would be doing if you were in the lab, you would be testing these things. Potato, bread. Now, a funny story about bread. For this lab, since I wrote them, I had to provide some of the chemicals because COD couldn't, not COD, ECC can't buy them through suppliers we normally go, and they're not going to give me money to go to Jewel or Myers. So I would bring it in. Now, because I'm Jewish and I'd bring in stuff and I'd like to sit a while and not go moldy on me, the bread I bought was matzah, which is starch. Well, I gave it to Dr. Daly, who's the lab manager. He made these nice bottles, and he wasn't thinking, and I wasn't either. And he put on the label matzah. And the first class I used, 5 minutes into the lab, a couple students came up to me and said, Dr. White, where's the bread? I said, oh, there's matzah here. What's that? I realized, oh, you led a sheltered life. And Dr. Daly changed the label after that lab, bread, bracket underneath, matzah. All right, now, I also had to bring in the cheese, flours, and Dr. Daly brought in the cereal, sugar, honey, cornstarch we had. They could buy. I don't know why they couldn't get cheese, but they couldn't. Well, one time, a while back, oh, the lab. So I went to my favorite supermarket, Valley Produce in Hoffman Estates. I don't live far from there, and I like going there. And they had on sale, the inexpensive sliced cheese, American cheese. And oh, okay, I bought it. In the lab, the student said, this is turning blue. I said, no, cheese doesn't have any starch, until I picked up the label of the yellow cheese that was on sale because it was cheap, and it had starch filler. And they found that. So I also, next lab after time I taught it, I got some more expensive white cheese, and you can see the difference of filler or not. And so you do all this. And then you have to answer these questions. And with that, I'm done for the day. I have to send out the password for test number 4 in a little while, but I'm done. If you have any questions, remember tonight I have my office hour. If I don't see you tonight, have a great rest of the weekend. Good luck on the test. Hopefully you won't need luck. And don't forget to use the method for those who know how to use the method. And with that, I'll say, gang is, oh, one thing before I log off, by Sunday, if not earlier, I will post in D2L your test 4 scores, and I will also, before Sunday, email out individual emails with the points you got for each answer you put down on test number 4. And then on Monday, I will go through the answers of test number 4, but I'll cut it out of the video. I like my special effects. And I'll cut it out of the video. And another thing I'll do is on Monday, I will show you, based on your 4 test scores, and assuming you've handed in all the labs, if you did, everybody got pretty decent, really good scores, as the labs aren't that hard. And I will show you how many points you need to get on the final to get the grade you want. And with that, I can now say, gang is on. Goodbye now. I stole that from Granny to Beverly Hillbillies. Bye.